Naomi did her undergraduate work at Stanford. She was interested in anthropology and also got involved in geology. And sort of following that interest, she went and did a master's degree with Jay Quaid at the University of Arizona. And there she started working on some of the sites in East Africa, a site called Gona that has been a very important site in the Afar Valley of Ethiopia. I had the privilege of going to the field with her in Ethiopia, and one of the things that I learned was she was very puzzled one day. The Ethiopian script is very different than the Roman script, and so we had a hard time reading the signs. And Naomi, was trying to struggle and read this Amharic script, and she suddenly realized, oh, I'm reading it backwards because it reminds me of Jewish and the Hebrew script. And so trying to read the Ethiopian script backwards, the words weren't making sense. But when she would read them correctly, she could read them. So after having finished her master's, she came to University of Utah, where she did a PhD with me, again, working in East Africa. And we did field work with her both in Kenya and in Ethiopia. We had a memorable trip to the Omo Valley with Frank Brown, and I did see a preview of one or two of her slides, so I think she's going to talk a bit about that. What I remember on that trip was spending 16 hours in one of the flattest places I've ever been to, 7 hours trying to get out of a mud puddle going towards the Omo, and then a week later, 9 hours in exactly the same place where we were thoroughly and irrevocably stuck. But we did get out, otherwise we would both be there today. After her PhD with me, she went to Caltech, working with John Eiler. She started there working on oxygen 17, and she's been following that isotope of oxygen around the planet ever since. She was at Johns Hopkins for several years, and now she is a faculty member at the University of Michigan. That's what that M stands for on the screen that you're viewing. So take it away, Naomi. Tell us about O17.
All right, yes, sir. Thanks, Terry. Thanks for the introduction, and also thanks, Gabe, for the invitation. It's really good to be back, but man, I need another invitation because this doesn't replace the Salt Lake City trip. So, but it's good to be back and to be in everybody's midst again, and to sort of be coming back to my roots. And I'm excited to tell you about what I've been doing. I've really been sort of tracking something down since I've been, really this has been, I feel like I keep answering the same questions. Actually, Gabe was a postdoc when I was a graduate student. I feel like I'm asking, we shared an office for a little while, and I feel like I'm still asking those same questions, which is either good or bad, but I certainly have more tools about it, and I think we've made some progress. So a lot of the things that I've been struggling with or trying to understand and constrain is how can we do more with the oxygen isotope records that we have, and specifically from terrestrial archives. And this has really been driving a lot of my research. Specifically, I want to know what additional stories about climate and hydrology, ecology, tectonics could we tell if we could better constrain these Delta 18O records. So as many of you know, we use them very widely, and they're sensitive to many, many, many things, which makes them in some ways fabulous, but in some ways horrible as well. And so the question that I have is, what can we do a better job of this? And can we use them as better tools? So as Turi mentioned, I got my start at working at the U of U, and this sort of embodies a lot of my PhD, going through the mud, carrying a heavy pack while Frank Brown was in front of me, just like, very casually walking and leading the way. But this was from that same eventful trip We intended to go when it was not rainy, but as Turi indicated in his introduction, it was rainy. We got stuck because there was water in vertisols and shrink swell soils. And as you guys know, when water gets into those silicates, anyway, it expands. So we got stuck many times. So this is sort of me tracing down Frank, and I just want to thank, this was actually my committee at Utah. So in addition to Frank and Turi, Jim Ellringer and Ed Zipser, and then Jay Quaid was also on my committee, and he's an alum too. So I thought I'd put him in there as well. Okay. All right, so this is how I sort of started with these questions, and this has motivated a lot of my work, is what drives oxygen isotope variation in soil carbonates in Eastern Africa. And this is something that I was doing. This is actually from part of my PhD, and we are initially focusing on carbon isotopes. This is from the Turkana Basin, where Turi's worked a lot in, and over the past, in the Pleistocene, there's an increase in carbon isotopes that is indicative of the sort of the final gasp of C4 plants really taking over as floodplains in Eastern Africa. We see that shift also in the Awash Valley and the Afar. What was really interesting, so this is in carbon isotope space, but in oxygen isotope space, we see that two different things are happening in different basins. So in the Afar Basin, there's not very much of a change, and in the Turkana Basin, there's a real change. So there's a real increase in delta 18O. So understanding what this change was and driving it has been really motivating me. How can we better understand these delta 18O records? So simply, when we think about delta 18O in the geologic record, and specifically in carbonates, you can think of calcite as a function of the oxygen isotope composition of temperature and the oxygen isotopic composition of the water from which it formed. So let's say that's lake. And then the lake water itself is a function of the isotopic composition of the water coming into it, and then evaporation once that water is in that basin and in that bathtub. And then also, of course, that the oxygen isotope composition of precipitation, as many of you know from particularly Gabe's work and others in the department, is highly sensitive to a variety of different climate processes. Okay, so if you really want to do paleoclimate with this, you need to constrain all of these things. You need to, depending upon which variable you're interested in, you need to constrain the others. Okay, so it's sensitive, but it's almost too sensitive. But we know that oxygen isotopes, and I won't spend too much time on these, but this is an example from a basin in Peru, so the Peruvian highlands, which is actually, I'm continuing to work in here, but we see variation in oxygen isotopes over the Holocene that people have explained by a combination of regional climate change and evaporation. Many of you know that this has been widely used to understand and reconstruct monsoons in speleothems. This is from looking at the East Asian monsoon intensity. If you're interested in more deep time, oxygen isotopes have been used in SHIRTs to understand large-scale global fluxes in oxygen and the oxygen balance, and weathering rates and basalts, and sort of really, and seawater temperature. So these are large-scale questions that oxygen isotopes have been used for. And this is a region where I'm starting to work also. This is from soil carbonates from the Central Andes. And this is an example where soil carbonates, in some ways, oxygen isotopes are used an uplift to get at this part of the story. They're lowering, and so this is inferred as uplift here, so uplift in the Altiplano, but the soil carbonates in the Atacama are interpreted as evaporation. And this is classic. So it's like, depending upon which story you want, you can interpret soil carbonate delta 18O in different ways. Okay. So what I want to do with this talk is I want to go over the basic, just give you a little insight in terms of how triple oxygen isotopes work in water, showcase some of our work in modern lakes and soils in teeth, and then consider the potential for using this to tackle paleoclimate problems, and tell you why this is so exciting. And you'll sort of see that this is truly like in Turi's footsteps. I basically, no material is sort of off limits, and in order to really understand something for the paleoclimate, well, we really need to understand how things work today. So I've spent a good part, I would say, of the last 10 years trying to understand the system in the modern waters and modern materials, and along with a lot of other colleagues, this has taken a lot of work to do, but I think it's finally game time, and we're starting to apply this to the geologic record. So our predictions are showing that it works and it's sensitive. So we're really, I guess that's the best way to do it. It is game time. These are some of the people who I've been working with. This is not a complete list, but a lot of the people up here were at the lab that Ben Passy and I, also a Utah alum, have ran at Johns Hopkins before we moved to Michigan. Some people you notice I haven't updated Leslie's affiliation, but Leslie Chesson was really involved in the early days when I was working with Shuning to understand how 17O is varying in tap water. There's varying water data sets that I've spent a long time on that these people contributed to that I won't talk about today, but you'll sort of see it in various parts. I'll present some work from Emily Beverly, who was a postdoc at Michigan and who's now a professor at University of Houston. Some of this work in terms of with Don Rodbell and Pradiga, Polisar, and Mark Abbott is also with my current student, Sarah Katz. We're starting to think about this 17O in lakes. Anyway, so this is, and Turi's here because we have 17O in tooth enamel, which we developed with my former student, Sophie Lehman. So I'll talk about all this. There's many more people involved here, but I want to emphasize that to sort of really understand this new system, I've had to collaborate with, and it's been an honor to collaborate with lots of different kinds of people with different expertise. Okay, so why bother doing this? Why have I gone to all this trouble just the past 10 years, just to the point where I feel like we can start to put it to use? But the reason is there's just a lot of Delta 18O records that are used for paleoclimate reconstruction, but there's too many degrees of freedom in the system. And in order to squeeze more information out of the geologic record, we need to control this variation. And specifically, something that I'm interested in controlling the influence of evaporation and other kinetic effects. And so the bottom line, something that you can take from this talk, is the 17O distribution is really sensitive to kinetic effects. And this is what we're going to try to capture. Okay. So we have a need in the paleoclimate system to do better. We also know that we can actually, this cap 17O distribution, this capital delta 17O, which I'll describe and define for you, small variations in its distributions can be measured, and it's sensitive to variations in the hydrosphere, okay? And now we also can make these measurements in the geologic materials. So there's been a lot of analytical advances to kind of get to this point, and so that we can actually use it. All right. So this is a classic plot by Mark Tiemens from 2006 in terms of thinking about this third isotope, which is quite rare, and this is why we haven't paid attention to it, because it's very rare, it's hard to measure. And if you think about it, this is delta 18O, and if you just look at this plot, -50 to 200 versus -20 to 120, basically 17O is varying, it's just half, it's varying half of what 18O is. So this is what's called the terrestrial fractionation line, TFL, and it has a slope of 0.5. And you can see that the stratosphere has a really distinct, stratospheric ozone and tropospheric ozone has really distinct this 17O species. So this capital delta 17O is a residual from an expected value. So it's how much you're deviating from off the slope. So this is basically the intercept, okay? You see various forms of oxygen have different kinds of fractionation, and all of these deviations are meaningful and they're very interesting to pursue. There's sulfates that are distinct. Here's ocean water here, so 0, 0. And then here's atmospheric water. And that gray ellipse, everything that I'm going to talk about is in that gray ellipse, okay? So there's been a lot of work that's been done outside of this gray ellipse, but really in the past 10, 15 years, this is where the growth is in this field, and this is why it's so exciting, because basically before people just ignored that gray ellipse and said maybe some people still want to ignore that gray ellipse and say it's not worth going after, but I feel like it is worth going after because there's a lot of information of the variation within that gray ellipse, and this is where we're basically going to dive into. But the variation is small, okay? So I'll first talk about triple oxygen isotope basics, and then I'll talk about it in lakes and soils and in teeth, okay? So here's some basic terminology, and don't worry too much if you're an isotope person, dig in. If you're not, the basic thing that I want to get from you is I want to communicate to you so that you get is the same oxygen delta 18O, which is the 18 to 16O ratio, relative to the 17O to 16O ratio. And in most cases they vary. This would be the TFL. They vary along this line, this line would be 0.5. Okay. So, but the important thing to notice is this 0.5. This equilibrium fractionation processes, so things that like distillation or any equilibrium fractionation would occur in the delta 18O space, it would occur along this blue line. Kinetic fractionation would occur along a smaller slope, a slower slope. And so this would be this red line. And this deviation from any sort of expected slope is your capital delta 17O, okay? And I'll leave it at that. Some people call it 17O excess, sort of like a D excess for in the hydrogen oxygen isotope space, or this capital delta 17O. What's important to realize is that these deviations are really small. So the differences in slope are very small. So this residual that we measure is also very small. So when I speak about these values, usually we present delta values in per mil notation. I'm going to be speaking about these deviations in per meg notation. So that is 0.001 per mil, okay? And so that's the way we'll talk about it. And let me just give you a sense of a little bit how this works. For those of you, I'm going to start this in terms of thinking about this in delta 18O versus delta D space. And when we first set up the lab at Hopkins and I tried to get this measurement to work, I really wanted to kind of see how just an even simple evaporation system could work. So this is just that same setup, delta 18O versus, in this case, delta D. There's also an equilibrium fractionation, which has a higher slope. In this system in waters, it's usually around 8. And then in the lower slope, kinetic fractionation can be anywhere from 4 to 6, okay? This deviation, this residual in delta 18O versus deuterium delta D is called the D excess. So we did an experiment to see if our measurements were working. And so here what we see is that we see fraction remaining. So we start out with a little pan of water and we evaporate it. As you evaporate it, as you guys know, that when you drive something to evaporation, the oxygen isotope value of it increases. But if you're going along this kinetic fractionation, your D excess should decrease. And that's what happens with water. Your starting water was here. This was Baltimore water. This is not Salt Lake City water, but this is Baltimore water. And as you evaporate out, your delta 18O values increase and your D excess values decrease. These hatch lines are just an example of just a very simple model that we did to sort of see if we are getting values that were reasonable. Okay, so let's do the same thing for 17O to see how this works. Okay, so here I've just replaced everything with, I've replaced the delta Ds with delta 17O and D excess with cap 17O. So here the same thing, that if you're driving something to evaporation, delta 18O is increasing. But if you're changing your delta 18O on a kinetic fractionation, you should have a lower slope. And so this capital delta 17O should be decreasing. And so we saw the same thing. This is exactly what we should expect to see. So delta 18O is increasing as you evaporate, but your cap 17O, your capital delta 17O is decreasing. But note the difference in scale, okay? So before I had, this is D excess is in per mil, okay? So we went from, 10 to negative 80, but this next one in cap 17O, we're going from around 20 per meg to, let's say, negative 180 per meg, okay? So that's really negative 180 per meg is only 0.18 per mil. So these are really, we had to, the precision required is on a orders of magnitude different to sort of get this. Because these differences are so small, it's actually really hard to see the differences in slopes. What we end up doing is we never actually end up plotting delta 18O versus what we call little delta 17O because you can't see it with your eye, okay? So instead, we plot delta 18O versus this residual. So we just rotate the plot, okay? So this means that if you have any fractionation that's equilibrium fractionation, it should be a straight horizontal line. And if you have fractionation that's kinetic, it should dive off along a slope like this, okay? And so I can plot the data like that. So this means that when you start with your pool of water here and you're evaporating it out, so you're increasing your delta 18O value, if it was all equilibrium fractionation, you would just have a horizontal line here. It would just look like that. But because it's evaporations, it's tailing off, right? It's taking that slope, okay? So this is sort of the vision of what we see when we see evaporations driving some sort of variation in delta 18O, the cap 17O, as delta 18O increases, the cap 17O will decrease. All right, and these are just the slope. So an equilibrium slope would be, let's say, 0.528, and a kinetic slope is around 0.518. So that's the difference in slopes, the difference between 0.528 and 0.518, and that's what we're trying to capture. As you can expect, if evaporation is really tracking evaporation, you should see a correlation between D excess and cap 17O, and that's exactly what we see. So when evaporation is driving a system, D excess and cap 17O are when evaporation is driving fractionation, excuse me, these two are correlated linearly, and that's beautiful. So we see it when you do have basically the luxury to measure DXS and 17O, then you can see they're correlated. Okay, so this is the basic premise, but my question is, can we use this as a tool for paleoclimate research? Okay, and if you can crack this code in carbonates, like, oh my, there are carbonates all over the place. So there's a huge potential to use this and to really change how we do our work. Okay, so what do we need to do? Let's go to town on this, right? That's what I was sort of thinking. How do we do it? So the first thing is you need to be able to measure the CAP 17O in geologic materials precisely. So we need a precise measurement. Then we need to figure out how the CAP 17O and these materials record climate and environment today. And then step 3, apply to the geologic record and and go to town on it. So where we are in this research right now is we've done step 1, we've done step 2, and we're like about to embark on step 3. And I will be really talking to you about step 2 today. Okay, I'm not going to go into the details of all these methods, but I want you to know that if you can measure the delta 18O of something, you can measure the CAP 17O, and it might not be done already, but it's possible. And the main challenge of all of these different methods is many of these methods rely on measuring actually the mass spectrometry. They measure CO2 or they measure CO. These are gases that are a lot easier to handle. For 17O, you actually need to you need to measure O2 because with CO2, there's anything that has 17O, there's going to be an isobar with 13C, and that's going to overwhelm your signal, okay? So this has been the challenge, to basically modify our methods and do things precisely enough to get to get down to it. But with all of these methods, with carbonates, silicates, oxides, bioapatite, and gypsum were there. Cellulose, it should be possible. And Ben Passy at Michigan, he's very close to getting they're just with his student Nick Ellis, they're working on actually looking at 17O and phosphates on the TCEA. This is for those of you who are the isotope nerds, and this also should be easily transferable to cellulose. So anyway, so that's where we are on that. But I'm going to talk about carbonates today, and the methodology is there. I'll also mention that some people are doing this work on cavity ring down instruments, and so that's a whole. I'll talk about mass spectrometry, but there's a whole level of ingenuity and technological advances that is happening to make these measurements. Okay, so because of this, what I'm going to go and get into is that, to me, kinetic effects, evaporation, I think aridity. And I think about that because that's something that I've been querying and trying to understand for a lot of my career. And this is really why I got into this. So I basically I started investigating this because, like, man, maybe this is what we can do and use to constrain aridity and evaporation on the record. So easy targets for this, things that we know have oxygen isotope records that are sensitive to aridity, are lake carbonates. Okay, that's an obvious one. Soil carbonates, those are also susceptible to aridity. Tooth enamel, and then eggshells. So these are like the easy slam dunk kinds of things. And when you're doing anything that has a hard component, well, you might as well start working with the materials that you think are gonna yield the most results the fastest. So this is what we've started with. I'm gonna show you this work in lakes, and I'll mention, I haven't put this in this talk, but I want to mention that if any of you guys are curious about 17O and want to know more, the Ilya Bindeman and Andreas Pack have organized a workshop at the end of AGU on December 18th through a volume that's in the Reviews in Mineralogy, so through the Geochemical Society. So it's a whole day of talk, and I'm going to obviously presenting work that's focused on my group, and but there's a lot of different workers and people doing this kind of research, and so it would be a great way to get an insight into all the latest and greatest. So this is a very biased view of just sort of my take on it. It's not a full review. So 17O in lakes. This is a paper that Ben Passy came out with his former PhD student from Hopkins, Haiyuan Ji, and what Haiyuan did, it was basically a first, we sort of get a first pass when you're new in a method, and he basically went around and he just did a grab bag of samples in terms of what were near modern lake carbonates and lake waters, and then also incoming waters into the watershed. So you can see that he went to the Great Salt Lake, he went to Bear Lake, Pyramid Lake, Lake Tahoe, and Mono Lake, okay? And then sampled waters and carbonates as many places around, all right? So to see what would happen. So what I'm plotting here is I'm plotting delta 18O, or what Ben plotted, is delta 18O versus capital 17O. And so you can see the inflow river water for Pyramid Lake, for Bear Lake, for the Great Salt Lake, for Mono Lake. These are all in the upper left-hand quadrant of the plots. And then what you have is you've got the lake water in this tan color, and then carbonate parent water. So this is carbonates, isotopic composition of carbonates that were measured for Cap 17 and then parent water has been reconstructed back from them. So this is all in water space, but those gray circles were actually started as carbonates. So the carbonate water, the reconstructed parent water for the carbonates, and the lake water, they all fall in, below and to the right of the incoming lake water. So this is incoming catchment waters, the inflow water. So they're all evaporated, which is exactly as you would expect. And they all follow slightly lower slopes than equilibrium. So they're all evaporated. They indicate kinetic fractionation. So this is exciting in some ways. So you can basically have a relationship between, you know, of course, in waters, you can measure this today with D access and delta H, you know, versus delta D, but we don't have D access in the geologic record. So from here, you can measure using the Cap 17. This is showing that you can measure the delta H and the Cap 17 of a carbonate and understand if how evaporated that water was. Okay, so if the carbonates came out, if the reconstructed waters came out up here, you would know that that lake is not evaporated. Without water, yeah, the water from which that formed is not evaporated. But Ben wanted to take it a step further, and he also wanted to then think about, okay, how can you solve for, how can you use those carbonate values, those reconstructed values from carbonate to kind of solve for the green diamonds? Like, how can you get back? Because that's also what we want. Not only how evaporated is something, but what was the initial water that was coming into the system? Okay. So we used a Monte Carlo approach and an isotope mass balance model to solve for the oxygen isotope composition of unevaporated precipitation. And taking this, these were the measured values on the x-axis, the measured values of the catchment waters. And then this is what you would sort of, the values up here above the line would be the evaporated versions of them. And then he reconstructed a reconstructed delta H value of them using that Monte Carlo approach. So this is exciting because you can basically think about, all right, you can reconstruct what the isotopic composition of the lake water was, and then take that and also consider what the initial river water and catchment water was. So this is a major advance into thinking about these systems. There's different ways that you could apply it. This is actually thinking about South America. So there's lots of obvious sort of roots to thinking about South America, a very arid part of the world in the Atacama specifically. So this is unpublished data. This is sort of replotted from some of the work from Phoebe Aaron, who's a recent PhD and now postdoc at U of M, who's worked to a certain degree. We've sort of adopted her in the lab. She has included 17 in her research. So this is with elevation, and you can see that rainwater doesn't change with elevation in Cap 17, but you can see that rivers and evaporated lakes do. This is some work done by other groups, by Surma and Hurwitz, looking at the Atacama Desert, and there's huge amounts of variation in ponds in the Atacama, which you would expect. But what's also really cool is they're also seeing these really low Cap 17 values in gypsum as well. And for comparison, this is Pyramid Lake, and this is how evaporated Pyramid Lake is. And of course, the salars in the Atacama are even more evaporated. But what I want to tell you is that this is sensitive, okay? And so as we start to look at this in other modern systems and connect waters to carbonates, we're seeing that there's a lot of traction here, and that the triple oxygen isotope of the carbonates give a faithful recording of what's happening in those evaporated basins. And so it's a really exciting way to get at paleohydrology that I think will open up a lot of frontiers to think about how we approach some problems. So this is just a little plug. There's lots of ways that we're working on this. I'm not going to steal the thunder of my student Sarah Katz, who's been doing this work in Lake Hunin, but she's presenting, for those of you going to AGU, she's presenting an AGU talk. This is just a screenshot of her slide. So she's been working on this in another lake basin, but Lake Hunin up in the highlands of Peru, and doing a really nice study in terms of looking at waters and carbonates, looking at clumped isotopes and 17O of this system, and showing that it also works in that system. And she's compared closed lake basins and through flow systems, so more open systems, and shown how this partitions hydrologically really well. So 17O is very sensitive to that. So we're getting ready to also dig into this core that was just drilled at Hunin too, but we first wanted to do the modern sort of template. Okay, so that's lakes. Lakes are an obvious target. There's tons of potential, and there's a lot of work that's being done on that. I couldn't pursue 17 without also thinking about soil carbonates. Soils are more complicated than lakes, but they are near and dear to my heart. And this is how this is actually what was really motivating me. That record from Turkana and why there was a huge range in 18.0 in Turkana really motivated me to even get into this business. And a perfect person to show up was Emily Beverly. So she's has a background in looking at paleosols, some isotope work, but really she was a paleosol expert. And so she came and she got an NSF postdoc to work with me at Michigan, and she did this phenomenal study of a transect of modern soil carbonates in the Serengeti to start to put this to work. So this is a basic model of Emily's in terms of just a schematic and thinking of the oxygen isotope composition of soil carbonates that form from soil water, which can be susceptible to evaporation and also plant dewatering. And so this is just a way of how do we think about the oxygen isotope composition of soil carbonate, that delta 18O SC, and soil water, and how do we make sense of it? Okay, so she did a study of a transect from the Serengeti, and this was actually important that she did. This was a place, a relatively small area to work with, but she was able to find, it's all grassland, so she was able to keep vegetation constant, and she was able to look across a precipitation gradient in the Serengeti and sample soil carbonates across this. Despite a lot of work that has been done in Eastern Africa in soil carbonates, we actually have an embarrassingly few number of studies, systematic studies of modern soil carbonates forming in East Africa. So one of the motivations there are many motivations for this, primarily thinking about 17O, but also just getting more data from modern systems in Eastern Africa that we just didn't have. And Emily did this in spades. So in a very short time period, she designed this beautiful study of going from from wet, so higher, this is Lake Victoria right here, so higher precipitations in the northwest, and then going a transect to more drier regions in the southeast of her study area. She collected soil waters. She did not collect hippos, but she collected surface waters, and then also soil carbonates across this gradient. And then she also measured, I won't talk about this data, but she also measured, she set up a bunch of soil temperature loggers, so she's got a beautiful soil temperature record, and also she collected tons of samples for organic matter, and actually Sarah Feekins and her students are doing a really nice leaf wax study, an organic study across this transect, and there's a lot more that can be done with this data set. So this is the transect and oxygen to carbon isotope space. So carbon isotopes on the x-axis and oxygen isotopes on the y-axis. And the first thing you'll see is that everything is very high in delta 13C, as it should be. This is a grassland. This is how Emily designed this study. She also plotted the other sort of modern or near modern soil carbonate data from Eastern Africa. There's not much from Eastern Africa. These are just the black dots. These are the only other modern soil carbonates that we know of from Eastern Africa. And so this is why they're distributed this way. But holy cow, just within the Serengeti, look at the variation in delta 18O that she sees. So anywhere from -5 per mil to +4 per mil. So these are all modern soil carbonates across this transect in the Serengeti. So there's a huge amount of variation that she's observed over a small period, a small space. Okay. So what she did here, this is actually using a tool that Gabe developed, the online precipitation isotopes calculator, whatever, maybe I'm butchering what that stands for, but basically there's not very many recorders of precipitation delta 18O in this region. So Gabe's tool was really useful in terms of understanding what local oxygen isotope values of precipitation would be. So this is expecting what we would sort of expect the oxygen isotope composition of the meteoric water, the local water would be across this gradient. And this is a calculated soil water on the y-axis of what we think soil water would be based on clumped isotope temperatures that we have, okay? And this is for all of the soil carbonates that she sampled, and this is where this huge range. So it seems like at the driest sites, so off in the southeast, she's got the most deviation of soil water, calculated soil water values from what she would expect based on local rainfall or the interpolation of local rainfall, okay? And so she's expecting, it's up to, more than 7 per mil above what you would basically predict or expect, okay? And so the hypothesis, right, is that this huge amount of variation in delta 18O of what you would expect would be due to evaporation. So this was looking promising when we saw this, because there was no going back and doing multiple seasons on her short string of a postdoc. So then what Emily did, and she took this, and from a handful of those, these analyses take a really long time. So she picked a handful of these samples. So, and these colors didn't come out on this slide, but you can see them on here. So the blues are the more mesic, more higher rainfall, and the warmer colors are the lower rainfall amounts of these different sites. And so you can see here that the blues and this yellow triangles are fitting on closer to this meteoric waterline. So remember, if there's no isotopic fractionation, then you're gonna see a horizontal distribution. And if you see kinetic fractionation, so this would be equilibrium, sorry, equilibrium would be horizontal, and evaporation or kinetic effects would fall below this line. So of the initial data set that we have, we see that the samples that are from these two sites, so from Shifting Sand and Malamba Road, so that these red squares and these orange, what are those, diamonds, those have the lowest CAP 17O values and the highest delta 18O values. So this is showing that this is actually what we predict, where I'm showing these triangles that are sort of cascading down are expected different, you know, if you sort of take different fractionation slopes from different starting waters, what you would expect for CAP 17O coming off of this. So this is really promising to think about. So it seems like we've been able to explain that, yes, evaporation is driving these. This is an initial pass. But another way to look at these is to think about, this is the data plotted in two different ways, to plot this capital delta 17O data as a function of aridity. So you can plot that in two different ways. You can look at it as MAP over M over PT. And so that would be an aridity index, or you can think of it in terms of water deficit, which would be PET minus MAP, okay? And so what we're seeing is that you have higher aridity, or this in the semi-arid, you have lower CAP 17O values right here, and then in the Serengeti, the dry subhumid areas, you would have higher CAP 17O. So this is showing to have promise. Julia Kelson is another postdoc working at Michigan right now, and she's taking a more global approach and thinking about what this means. So I think we're on the brink. It's showing a lot of promise of that we're on the brink of really understanding how this system works. Okay, enough soils. So we're making progress there. My last vignette here is to think about this in teeth, which is another thing that I worked on with Turi, and obviously soils and teeth are not falling very far from the tree here in terms of having worked with Turi. And this work is with Turi in part, but it was led by a PhD student of mine, Sophie Lehman. And this was a main one of the chapters of her dissertation at Hopkins. And she is like almost maybe a week away from finally getting this out to co-authors. But it's a very exciting study. And one of Sophie's hypothesis was that she thinks that in more arid places, the CAP 17O of tooth enamel will be lower and more variable. So this is a plot from Sophie's paper showing meteoric waters and their distribution in delta 18O space and capital delta 18O space. These green, the green triangles are from plant water. This is mostly from Shuning Lee's data set that she generated from Impala, Kenya, that she generated while at Hopkins. Oops, I'm gonna go too fast. And this shows that this is an evaporation driven signal. And these diamonds, these pink diamonds, are body water, expected body water from measured from eggs from large birds and from mammals. So this is sort of a predicted amount of body water CAP 17O should be. Okay. So the ideas for this part, and this is what was motivating Sophie's study, is that the capital delta 17O of tooth enamel will reflect leaf water values, that those animals that eat leaves, and they obtain a large fraction of their water from leaves. With the idea that the capital delta 17 of an animal will be sensitive to aridity. And this range in capital 17 delta 18O values will also increase with aridity. So here's a basic way to kind of think about it. You can think about this top panel when we only had delta 18O in a place when there's not very much aridity, so a more mesic place where there's more rainfall. Let's say you have a hippo who likes to live in water versus a giraffe. The hippo and giraffe in delta 18O space are going to be pretty close together, okay? When you get to a more arid environment where the giraffe's going to be relying more on more leaves for its for its water intake, those leaves are going to be evaporated and there's going to be a greater separation in delta 18O space between the hippos and the giraffe. And this is actually something that I worked with Turi on to look at it, to develop an aridity index just based on delta 18O and on animals like hippos and giraffes, okay? But that concept, I mean, well, that was, I can say it myself, it's really limited, that approach, and in part because you need to know who your hippo is and you need to know who your giraffe is. So you go back in time in the geologic record and it's harder and harder to apply. And it's also harder to apply in different regions. So you can think in one region that your hippo and giraffe, let's say in Eastern Africa, where they have relatively high delta 18O values. But you can imagine that, let's say you're starting you start with in the Intermountain West, where you have bison and antelope or a deer, and you could easily imagine that this deer that's eating a lot of leaf water could be, or pronghorn antelope, could actually have exactly the same delta 18O value as that hippo from Eastern Africa, okay? But you wouldn't necessarily know that by just looking at the geologic record. You would have no way of knowing that one is very evaporated and started and is from a place with the oxygen isotope composition of water is very low, whereas one is not evaporated at all and the isotopic composition of the local water is very high. So if you add this CAP17O concept to it, you could separate out two regions, okay? So you would have this evaporation insensitive, like you would have your buffalo or your bison and your hippo, and they start in very different delta 18O spaces because they represent very different places, very different geographies. And then you have your comparative evaporative or more evaporative sensitive animal. So let's say your antelope or your giraffe, okay? And the idea of how this would work is then you can then put all of these things together. You could put in aridity, so as in less arid environments, most of your data sets are going to be all of your animals are going to be plotting high in CAP17O. And as you move to more arid environments, as you move to the right, you're going to have more of a spread in data, all right? And so when Sophie was thinking about this, she was predicting that these environments, you would basically expect a wedge-shaped pattern in your capital delta 17O of your teeth. And what's cool about this is you can scramble the buffalo and the bison, and you could scramble the hippos and the giraffe all together, and any other reindeer, and it doesn't matter on what your initial delta 18O value is. It doesn't matter on regional geography because this capital delta 17O is insensitive to that, all right? It just matters in terms of this overall space. Okay, so this is what we tried, and we carefully, since the analyses are really laborious, we carefully sampled samples from across the world, but very well chosen. So what I'm showing you here is all the samples in Sophie's data set, and what you can see is all the squares are from Africa, and they're by absolute latitude. So we have some from South Africa. So the darker squares are from South Africa, and those that are yellow are from closer to the equator. And then we also have data from the US and Finland, and including in the US is from Alaska as well, okay? So what you see here is in terms of, you're plotting delta 18O versus aridity, there's nothing, right? It's a big fat blob when you put all of these data together, okay? If you plot capital 17O versus aridity, aha, we see our wedge, okay? So these are less arid environments and these are more arid environments, and it's exactly as Sophie predicted, is that these more arid environments have a greater range in capital 17O, and there's just more potential for evaporation, and these less arid environments have a much smaller range in cap 17O. What's cool about this is you can see that the circles and the squares are totally commingled, and also the, except for the really high latitude sites, also the colors are commingled. So this is a way to get at aridity that's not dependent upon latitude or region, which is phenomenal. That's something that we really were stuck with the last approach. And here is plotting the cap 17O versus the delta 18O values, okay? So yeah, as you see in general, the lower cap 17O values, of course, plot with the higher delta 18O values, but it's not always the case. So this is a perfect sample point to kind of think about, whereas it sort of fits right in line in the cap 17O versus aridity space, but it seems anomalous here. So this is an evaporated sample, an arid place, but that is started in a low delta 18O world, okay? So that's what we see here. You can also plot the data in terms of taxon instead of by latitude. So you can think of, just if you're curious in terms of bovids or beavers or deer or elephants, giraffe, hippo, rhino. We have other taxa too, but you can see how these kind of parse out by taxonomic affiliation as well. So another way to think about this, we split it into evaporators and insensitive. So these would be the buffalo, the bison, the hippos, the elephants, and the sensitive taxon, so the antelope, the deer, and the giraffe. And if you look at this in terms of delta 18O space, there's not, and these are things that we didn't have like an expectation. They were sort of hard to classify. If you look at these in delta 18O space and you take this, not fully global, but representative global of data set from a diverse set of places, there's very little separation between these two groups. But if you plot them merely in the cap 17O space, you can really see clearly that these insensitive taxa, so these animals that are more water dependent, have very little variation in cap 17O, where the animals that are more water independent, like those giraffes that would be depending upon leaf water, are on average much lower, and they have a much lower range. So this question is, can we use this to track aridity? I think yes. And so we're really close to starting to use this. And so that's where I'll just sum up. I think this has huge potential, and we're starting to really lay the careful, slow groundwork to show how it works in modern systems, which has been taking a lot of work, but really satisfying and fun to uncover all of these, how the system works. And now it's a matter of finding really good systems to apply this and to show how what we can do with it. So I've shown you from soil carbonates, lakes, and teeth. Ty Huth, who's also a graduate of U of U, is at a postdoc in the lab, and he's starting to work in speleothems. I didn't get his AGU talk in here, but he's giving a talk at AGU, which is really cool results on speleothems. So we're sort of moving there. And as I mentioned, Ben's working on developing this better in fossil teeth and phosphates, so we can analyze it in smaller samples. And there's just tons of potential for this work. Also in tree rings, I think is going to be a potential. So this is some pictures of people in the lab. So this is Ty, who's working in speleothems, Julia Kelson, who's working in soil carbonates. Phoebe Aaron these are all postdocs or affiliated with the lab. She's working in waters and is going to come out. You'll stay tuned for a lot of cool studies based on 17O and waters. So that is that. And with that, I'd be happy to answer questions and about anything that's happening. So thanks for your time.